Good morning and welcome to the second edition of our Horizon case study series, where we discuss some of the variants we have encountered. My name is Marese Grehan and I'm the Genomics software specialist with GenomeArc. Today I'll be focusing on a causal de novo KMT2D variant and its implications in Kabuki Syndrome. So approximately 80% of rare diseases are genetic in cause, with an average time to diagnosis of roughly 5 years. This long journey to find answers can cause stress and strain for patients and their families. Our series today will also highlight the role rapid tertiary analysis and streamlined variant interpretation has in reducing time to diagnosis by accurately classifying variants based on ACMG guidelines. Today's case focuses on a pediatric patient who presented with multiple congenital abnormalities that remained unexplained with standard testing methods. Moving towards a definitive diagnosis, advanced genomic screening and genomic variant interpretation completed with GenomeArc Horizon were needed to identify this complex condition that traditional medicine could not resolve. So our 7-year-old patient presented with bilateral hearing loss, microtia and atrasia, along with eczema and developmental delay. Further evaluation identified a congenital hepatic cyst, resolved congenital heart disease and atonic seizures, each requiring evaluation and assessment by several specialists without a unifying diagnosis. The next step was genomic investigations to search for underlying genetic causes that could explain the patient's phenotype. The child, along with the parents, underwent whole genome sequencing to resolve the patient's complex clinical presentation and to understand the genetic inheritance patterns. So using trio analysis supported by our Horizon platform, enabled ACMG-based reporting of primary and secondary findings along with carrier screening and population-specific variant assessment. This workflow allowed us to efficiently move from whole genome sequencing to variant interpretation, providing a genomic explanation for our patient's clinical presentation. So at this stage, we moved from broad genomic data to evaluating which variants could actually explain the patient's presentation. So using ACMG criteria, we examined each candidate variant for strength of evidence, including functional impact, population frequency, inheritance, and clinical fit. The ACMG framework organizes variant interpretation into 5 categories: pathogenic, likely pathogenic, variants of uncertain significance, likely benign, and benign, based on disease causation and clinical significance. 28 evidence codes describe different levels of support, ranging from strong disease-causing indicators, like de novo occurrences or loss of function, to population data, computational predictions, and phenotype associations, providing validated evidence of variant pathogenicity. So the primary finding in our case was a de novo heterozygous stop gain variant in the KMT2D gene, which was identified through trio analysis of whole genome sequencing data from the child, father, and mother. So because this variant introduces an early stop codon, it can disrupt gene function, and its absence in population databases further supports clinical relevance. Applying ACMG criteria, the evidence met the thresholds for a likely pathogenic classification. Trio analysis also confirmed autosomal dominant inheritance, with the variant arising de novo rather than being inherited from either parent. This result could now provide an explanation for the patient's phenotype and was consistent with Kabuki Syndrome. So we could now connect the patient's clinical features to the underlying biological mechanisms of the KMT2D variant. So KMT2D encodes a lysine-specific methyltransferase, an enzyme responsible for adding methyl groups to histones. These help regulate gene expression during development. So in normal cells, KMT2D-mediated histone methylation ensures that key developmental genes are activated at the right time and in the correct tissues. So with this de novo loss of function variant, the KMT2D protein becomes non-functional, disrupting histone methylation and altering the expression of multiple downstream genes. So this disruption can affect embryonic growth and organ development, particularly in craniofacial tissues, the auditory system, immunoregulation, and heart development. So this explains the clinical phenotype of our patient, and findings were consistent with Kabuki syndrome. So along with the primary Kabuki diagnosis, trio sequencing revealed inherited HbA1 variants consistent with the alpha thalassemia trait. So unlike the novel KMT2D finding, these well-characterized pathogenic variants are known to reduce alpha globin production and met ACMG criteria through established functional and population evidence. Trio analysis showed that each parent contributed one variant with compound heterozygous inheritance. Although clinically subtle, this secondary finding carries meaningful implications for the family. So alpha thalassemia is an inherited blood condition caused by reduced production of the alpha globin chains that form part of the hemoglobin molecule. Variants in HbA1 can decrease the number of functioning alpha chains, which can lead to reduced hemoglobin synthesis and mild microcytic anemia in carriers. In this patient, the inherited trait is clinically mild, but important to recognize for future family planning, as having two carrier parents increases the risk of severe alpha thalassemia in children. So through the integration of trio sequencing, ACMG-guided interpretation, and inheritance confirmation interpretation with GenomeArc Horizon, the clinical team could now distinguish between a de novo primary finding and inherited secondary finding in our pediatric patient. This was further confirmed through IGV, providing a clearer picture for the family and actionable results for the clinician. The Horizon platform supported the clinician in making their clinical diagnosis and could also link the phenotype to genotype by automatically linking unstructured clinical notes to standardized HBO terms. This enabled more precise phenotype-genotype matching. Finally, in our case, the Horizon platform streamlined clinical reporting by automatically populating variant details for both primary and secondary findings, while still allowing the clinician to register their complete interpretation and integrate patient-specific information and any additional supporting evidence. So in our pediatric case, the genomic diagnosis provided the family with long-awaited answers and practical next steps. So with confirmation of Kabuki syndrome and identification of alpha thalassemia trait, they finally received answers and access to syndrome-specific resources and tailored clinical management. The de novo origin of the KMT2D variant offered reassurance to the parents regarding reoccurrence risk, while carrier information for HbA1 was also discussed. Together, these findings demonstrate how comprehensive sequencing, along with fast and accurate variant interpretation, can guide both medical care and the next steps for the family. So in this case, we saw the impact of GenomeArc Horizon in transforming whole genome sequencing data into a clinically meaningful diagnosis. So by integrating genomic VCF files with phenotype-driven analysis, the platform rapidly prioritizes potential variants using ACMG rules, clinical databases, and AI-supported HBO term matching. Its tertiary analysis engine supported trio interpretation, carrier screening, and secondary findings within minutes, and enabled efficient identification of the de novo KMT2D pathogenic variant and the clinically relevant HbA1 carrier status. The streamlined workflow facilitated accurate classification, automated reporting, and ultimately guided clinical decision-making. This case highlighted the importance of genomic medicine in rare disease diagnosis, where complex phenotypes create a difficult puzzle for clinicians in reaching an accurate diagnosis and possible pharmaceutical interventions or therapeutics if available. So as we reach the end of today's case study, I just have a few audience questions, and the first question is how much shorter the protein will be due to the stop gain mutation. So the mutation affects exon 40 out of 56, meaning the protein will lose almost 30% of its amino acids. So given that the gene plays an important role in histone modification and regulates numerous genes, the stop gain mutation is likely to result in a loss of function effect. So in our second question, we have, apart from ClinVar, what other databases does the Horizon platform use to support variant pathogenicity? So Horizon has its own proprietary databases that is curated around 100,000 variants of clinical relevance. So this includes pathogenic, likely pathogenic, and variants of uncertain significance. So these variants are independent of ClinVar, and Horizon updates its dataset every 3 to 4 months. So thank you for registering for the GenomeArc case series, where each month we present a new case on the variant interpretation. I hope you found the presentation of our pediatric case study involving the de novo KMT2D variant interesting. If you would like any more information on today's discussion or on how we use Horizon to classify variants, then please reach out and I will be happy to discuss further.